Hello everyone. Today we will discuss about HMP Shunt. This is an alternative pathway for oxidation of glucose. Other names of this pathway are Hexose Monophosphate Pathway, Pentose Phosphate Pathway, Pentose Cycle, then Phosphogluconate Pathway or Warburg Dickens Lipman Pathway. So this is an alternative pathway for oxidation of glucose. So let's see the metabolic pathway. This reaction in this pathway occurs in two stages. Reaction occurs in two stages. Stage 1 and stage 2. In stage 1, this is known as oxidative phase. Stage 1 is oxidative phase. This is oxidation of glucose and formation of pentose phosphates. Here in the stage 1, that is oxidation of glucose and formation of pentose phosphates, that is oxidative phase. Stage 2 is non-oxidative phase. That is here pentose phosphate reactions, that is interconversion of pentoses. Interconversion of pentoses and conversion of pentose phosphate to hexose phosphates, that is in stage 2, that is non-oxidative phase. So first we will see stage 1, that is oxidative phase. So stage 1 is oxidative phase. Oxidative phase means oxidation of glucose and formation of pentose phosphates. So first glucose 6-phosphate by the enzyme glucose 6-phosphate dehydrogenase, that is G6PD. Glucose 6-phosphate dehydrogenase, it is converted first to 6-phosphogluconolactone. 6-phosphogluconolactone. This G6PD here NADP plus NADPH plus H plus. Here Mg plus plus, Mn plus plus or Ca plus plus. Then the 6-phosphogluconolactone by the enzyme gluconolactone hydrolase. Gluconolactone hydrolase, it is converted to 6-phosphogluconate. 6-phosphogluconate. Here hydrolysis, so water, it needs Mg plus plus, Mn plus plus or Ca plus plus. So in the first step, this glucose 6-phosphate, it is converted to 6-phosphogluconolactone, then to 6-phosphogluconate. This reaction is, first reaction is catalyzed by the enzyme glucose 6-phosphate dehydrogenase or G6PD. It is an NADP dependent enzyme. Then this hydrolysis of this gluconolactone, 6-phosphogluconolactone, is by the enzyme gluconolactone hydrolase and it forms 6-phosphogluconate. This G6PD, that is glucose 6-phosphate dehydrogenase enzyme, is a dimer or a tetramer. It is the rate limiting enzyme in the pathway. That is G6PD, that is the rate limiting enzyme. Glucose 6-phosphate dehydrogenase is the rate limiting enzyme in the HMP shunt. This is the first reaction. Then the second step in the oxidative phase is this, that takes place in two steps. First step is this 6-phosphogluconate is converted to 3-keto 6-phosphogluconate by the enzyme phosphogluconate dehydrogenase. So by the enzyme phosphogluconate dehydrogenase, phosphogluconate dehydrogenase, this 6-phosphogluconate is converted to 3-keto 6-phosphogluconate. 3-keto 6-phosphogluconate. It is converted first to 3-keto 6-phosphogluconate. That is the first step in the second oxidative stage. Here NADP plus act as the hydrogen acceptor. Here also NADP plus NADPH plus H plus. NADP plus NADPH plus H plus. Here also Mg plus plus, Mn plus plus or Ca plus plus. This dehydrogenase enzyme, it is also a dimer, that is it consists of two identical subunits. That is the first step. Then second step is this 3-keto-6-phosphogluconate undergoes decarboxylation. It undergoes decarboxylation, that means carbon dioxide is released. So here in the HMP shunt there will be formation of carbon dioxide. So second reaction is 3-keto-6-phosphogluconate undergoes decarboxylation and forms a ketopentose, that is D-ribulose 5-phosphate. D-ribulose 5-phosphate. So this 3-keto-6-phosphogluconate undergo decarboxylation and forms a ketopentose, that is D-ribulose 5-phosphate. So this is the first stage. Up to this is first stage. Then in the second stage is non-oxidative phase. So in the non-oxidative phase there will be pentose phosphate reactions, that is interconversion of this pentoses, that is this D-ribulose from the D-ribulose 5-phosphate. A variety of pentoses are formed, that is the second stage. So here this D-ribulose 5-phosphate is acted upon by a ketoisomerase. It is first acted upon by a ketoisomerase by the enzyme ketoisomerase. This D-ribulose 5-phosphate, it forms D-ribose 5-phosphate. D-ribose 5-phosphate. This D-ribulose 5-phosphate is acted upon by ketoisomerase and it forms D-ribose 5-phosphate. This D-ribose 5-phosphate again can be converted to D-cellulose 5-phosphate. D-cellulose 5-phosphate. So this D-ribulose 5-phosphate, it can be converted to D-ribose 5-phosphate or D-cellulose 5-phosphate. This formed D-ribose 5-phosphate can be converted to D-ribose 1-phosphate and D-ribose 1,5-diphosphate. So it can be converted to D-ribose 1-phosphate and D-ribose 1,5-diphosphate by the enzyme phosphotransferase. Enzyme phosphotransferase, it is converted to D-ribose 1-phosphate and D-ribose 1,5-diphosphate. Here one molecule of ATP, ADP, Mg++. That is the reaction. Then that is the first reaction in this D-ribulose 5-phosphate, it is converted to D-ribose 5-phosphate and D-cellulose 5-phosphate. This D-ribose 5-phosphate can be converted to D-ribose 1,5-diphosphate and D-ribose 1-phosphate. Then this D-ribulose 5-phosphate which is converted to D-cellulose 5-phosphate. This cellulose 5-phosphate is another ketopentose. This is by the enzyme epimerase. Here it is ketoisomerase, here it is epimerase, and it is in the formation of D-cellulose 5-phosphate. Then some of the ribose 5-phosphate formed this ribose 5-phosphate, they can be epimerized by phosphopentose epimerase and it forms arabinose 5-phosphate. This formed D-ribose 5-phosphate, they can be epimerized by phosphopentose 2 epimerase. By the enzyme epimerase, they form arabinose 5-phosphate, arabinose 5-phosphate. Then that is the first step in the stage 2. Then to the second step, conversion of these pentophosphates to hexose phosphate. These all are pentose phosphates. Ribulose 5-phosphate, then ribose 5-phosphate, cellulose 5-phosphate. These all are pentose phosphates. So next step is conversion of these pentose phosphates to hexose phosphates. In this reaction, there will be conversion of pentose phosphates to fructose 6-phosphate, which is then isomerized to glucose 6-phosphate. So glucose 6-phosphate means it can begin the cycle again. So this is achieved by the enzymes transketolase and transaldolase. And these reactions are called as transketolation and transaldolation. So coming to the reactions, this ribose 5-phosphate, from the ribose 5-phosphate, this can by the enzyme transketolase. Here transketolase. It combines with this D-cellulose 5-phosphate. Ribose 5-phosphate plus D-cellulose 5-phosphate. It forms pseudoheptulose 7-phosphate. Pseudoheptulose 7-phosphate. Plus, here it is glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. So, ribose 5-phosphate, it combines with the D-cellulose 5-phosphate and forms pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate by the enzyme transketolase. This transketolase enzyme needs thymine pyrophosphate or TPP as the coenzyme, and it needs Mg++. So, in this reaction, there will be conversion of these pentose phosphates to hexose phosphates, that is fructose 6-phosphate, which is then isomerized to glucose 6-phosphate and begin the cycle again. So here the two enzymes are there, transketolase and transaldolase, and these reactions are called as transketolation and transaldolation. So in the transketolation and transaldolation, in the HMP shunt there will be two transketolation reaction and one transaldolation reaction. In the transketolation reaction, this pseudoheptulose 7-phosphate, formed pseudoheptulose 7-phosphate, pseudoheptulose 7-phosphate, it combines with the glyceraldehyde 3-phosphate. It combines with the glyceraldehyde 3-phosphate and it forms fructose 6-phosphate and erythrose 4-phosphate. Fructose 6-phosphate and erythrose 4-phosphate. Pseudoheptulose 7-phosphate, it combines with the glyceraldehyde 3-phosphate to form fructose 6-phosphate and this erythrose 4-phosphate. Then this erythrose 4-phosphate, it combines again with this cellulose 5-phosphate. So cellulose 5-phosphate again combines with the erythrose 4-phosphate. Erythrose 4-phosphate and it forms fructose 6-phosphate again and glyceraldehyde 3-phosphate. Fructose 6-phosphate and glyceraldehyde 3-phosphate. So that is the reaction. Here transketolase and transaldolase reaction. Transketolation and transaldolation. This transketolase means it requires thymine pyrophosphate as the coenzyme. It converts the keto sugar into aldose sugar with 2 carbons less. So here in the reaction, this D-ribose 5-phosphate, it is converted to the pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate. So that is a transketolation reaction. Here it is transaldolation reaction. Pseudoheptulose 7-phosphate combines with the glyceraldehyde 3-phosphate to form erythrose 4-phosphate and fructose 6-phosphate. Then again a transketolation reaction. Cellulose 5-phosphate combines with the erythrose 4-phosphate and form fructose 6-phosphate and glyceraldehyde 3-phosphate. So here this cellulose 5-phosphate, that is a ketose, that is 5 carbon. It reacts with the ribose 5-phosphate, that is also a 5 carbon aldo sugar. This is keto sugar and aldo sugar. So it combines to form pseudoheptylose 7-phosphate, that is 7 carbon, and it forms glyceraldehyde 3-phosphate, that means 3 carbon. So it forms a 3 carbon compound. Then this pseudoheptylose 7-phosphate, that is a ketose, that is 7 carbon. It combines with the glyceraldehyde 3-phosphate, that is 3 carbon, and it forms fructose 6-phosphate and erythrose 4-phosphate. Fructose 6-phosphate means ketohexose. Erythrose 4-phosphate is aldo sugar. Then again in the transketolation reaction, xylulose 5-phosphate means it is a ketopentose. Erythrose 4-phosphate means it is aldo sugar. So it combines to form fructose 6-phosphate. Again a ketose, keto sugar, and glyceraldehyde 3-phosphate is an aldo sugar. So this is the transketolation and the transaldolation. So in a HMP shunt there will be 2 transketolation reaction. Here one, here is the second reaction. Then this reaction is aldolation reaction, transaldolase reaction. So this is the HMP shunt. Coming to the fate of this fructose 6-phosphate and glyceraldehyde 3-phosphate. This glyceraldehyde 3-phosphate, this formed glyceraldehyde 3-phosphate, it can be converted by the enzyme isomerase to form dihydroxyacetone phosphate. It is converted to dihydroxyacetone phosphate by the enzyme isomerase. Then this glyceraldehyde 3-phosphate can combine with the dihydroxyacetone phosphate and it forms fructose 1,6 bisphosphate. Fructose 1,6 biphosphate by the enzyme aldolase. Then by the enzyme fructose 1,6 bisphosphatase, it is converted to fructose 6-phosphate. Fructose 6-phosphate by the enzyme fructose 1,6 bisphosphatase. Then fructose 1,6 or biphosphatase, it is converted to fructose 6-phosphate. This fructose 6-phosphate by the enzyme isomerase, it can be converted to glucose 6-phosphate. This glucose 6-phosphate means it can recycle the pathway. It can recycle the pathway. So this is about the HMP shunt. Then coming to the regulation. So let's see the regulation of HMP shunt. So first step, that is catalyzed by the enzyme glucose 6-phosphate dehydrogenase, that is glucose 6-phosphate to 6-phosphogluconolactone. 6-phosphogluconolactone by the enzyme glucose 6-phosphate G6PD, glucose 6-phosphate dehydrogenase. Here NADP plus NADPH plus H plus. This is the first reaction. So first reaction is the rate limiting step in the pathway, catalyzed by G6PD, that is glucose 6-phosphate dehydrogenase. This reaction is regulated by cytoplasmic levels of NADP plus NADPH, that is NADP plus NADPH ratio. So this is regulated by this ratio, NADP to NADPH ratio. If the cytoplasmic ratio is high, that is a rise in this NADP plus means, this ratio will be high. So it enhances the rate limiting reaction and the shunt pathway. So decrease in the ratio means that is a rise in NADPH. If there will be a rise in NADPH, it inhibits both G6PD and the 6-phosphogluconate dehydrogenase by making less NADP plus available for the catalytic reactions, and also by competing with NADP plus to occupy the enzyme binding sites of this G6PD. Then activities of this both dehydrogenases, G6PD and this phosphogluconolactone dehydrogenase, phosphogluconate dehydrogenase, and the rate of the pathway are enhanced by feeding high carbohydrate diet. These are enhanced by feeding high carbohydrate diet, and these are reduced in starvation and diabetes mellitus. Then increase in fatty acid synthesis and steroid synthesis. Increase in fatty acid synthesis and steroid synthesis. These reoxidizes NADPH to NADP plus. So there will be more NADP plus. So consequently the ratio will be high. So it enhances the shunt pathway. Coming to hormonal regulation, insulin, it induces the synthesis of both dehydrogenases and so enhances the activity of the pathway. Then thyroid hormones. Thyroid hormones enhances the activity of G6PD and so the shunt pathway. Coming to the metabolic significance of this, these are the regulation of this HMP pathway. Then coming to metabolic significance of this pathway, coming to metabolic significance, here there is formation of NADPH. This formed NADPH in this pathway is used as electron donor in many reductive synthesis in the body. For example, fatty acid synthesis, cholesterol synthesis, then steroid synthesis, sphingolipid synthesis, and then in uronic acid pathway. So this NADPH are used as electron donor in many reductive synthesis in the body. Then second significance is supply of pentoses. Pentoses means ribose 5-phosphate, D-ribose. These are used for the nucleic acid synthesis. Then arabinose 5-phosphate. Arabinose 5-phosphate. This formed arabinose 5-phosphate, these are used for incorporating this arabinose in glycoproteins. Then role in RB cells fragility. RB cells fragility means HMP shunt in RB cells provide NADPH. This also produce NADPH. This is for the reduction of oxidized glutathione. Oxidized glutathione means GSSG. Oxidized glutathione to reduced glutathione, that is 2GSH. Oxidized to reduced glutathione. This is NADPH is used for the reduction of oxidized glutathione to reduced glutathione by the enzyme glutathione reductase. That is in RB cells fragility. Next one is role in lens metabolism. In lens metabolism, same way this HMP shunt provides NADPH. This is necessary to convert GSSG to 2GSH, which is necessary for maintenance of lens proteins. Then role in phagocytosis. Phagocytosis. Here the NADPH generated in this pathway is utilized by NADPH oxidase in producing superoxide anions. This NADPH produced this utilized by NADPH oxidase to produce superoxide anions for destroying phagocytosed materials. Then another metabolic significance is production of carbon dioxide. This carbon dioxide produced in this pathway may be used in carbon dioxide fixation reaction. So these are all about HMP shunt. So let's see the differences between glycolysis and this HMP shunt. Coming to the differences of this glycolysis and HMP shunt, this glycolysis, it can take place it occurs in all tissues. In all tissues. Here in HMP shunt, it occurs in certain special tissues. Special tissues for special functions. Occurs in special tissues for special functions. Then the HMP shunt is a multicyclic process. Multicyclic. This is not a multicyclic process. Then here hydrogen acceptor is in glycolysis, it is NAD plus. Here it is NADP plus. Then another difference is here ATP is produced. Here this is not not meant for energy and it not produce ATP. Then here in the glycolysis carbon dioxide is not produced, but here it is carbon dioxide is produced in the HMP shunt. So these are about HMP shunt. So this is today's topic. Thank you for watching.